All right, thanks very much for having me back here. And my task is to try and place in proper perspective what kind of treatments you can expect over the next 6 months to a year. As we all recognize, these are exciting times in the field of hepatitis C because of the variety of treatments that are potentially going to be available. Unfortunately, there's going to be some variability across various parts of the world in terms of having these treatments available over the next year. It may take longer in some parts of the world. So to that end, to just give you all, again, an overview, which you all have heard several times, hepatitis C is a progressive form of disease, and these patients can end up with cirrhosis and its complications, including liver cancer. And in terms of disease burden, it varies from region to region, but in North America, it's expected that over the next 10 years, we'll have an enormous disease burden due to cirrhosis and its complications. So there is a pressing need to try and treat these patients so that you can control disease progression. Also, we know that there are various genotypes around the world, and I'm just showing this because if you look at Brazil, you will see that there is a lot of genotype 3 in Brazil. Of course, genotype 3 is more prevalent in countries such as India and Pakistan and amongst IV drug abusers, but you have a sizable number of genotype 3 patients, it appears, in Brazil. And they are going to be the next challenge because you'll realize after my presentation that genotype 1 is not going to be much of a challenge, whereas 3 might be the next genotype 1. So you heard this several times that there are 3 main classes of drugs that work against 3 regions of the virus. You have the protease region, NS3A4 region, and then you have the NS5A region, and then you have NS5B region. So you have the protease inhibitors against the NS3A4 region. Again, you have the NS5A, and then the NS5B region can be suppressed by 2 categories of drugs, the non-nucleoside inhibitors and then the nucleotide inhibitor, which is sofosbuvir. And you have a variety of non-nucleoside inhibitors. So just to get a little flavor, you have to look at it like the palm of your hand. The non-nucleoside inhibitors attack the fingers and the thumb, whereas the NS5B nucleotide inhibitor attacks the center of the palm. So you have the protease inhibitors, telaprevir, boceprevir, and the more recently approved cimeprevir. And then you have the NS5A inhibitors, daclatasvir, ledipasvir, ABT267, which is been named ombitasvir. And then you have the next generation NS5A inhibitor, which is pan-genotypic, which is 5816. And then you have the pan-genotypic MK8742. So if you were to look at pill burden in hepatitis C therapy, you're going to be looking at it this way. Ledipasvir and sofosbuvir has already been co-formulated and has completed phase 3 studies. 5172, a protease inhibitor, and 8742, a pan-genotypic NS5A inhibitor, is in phase 3 studies as a co-formulated pill. Then you have regimen of ritonavir boosted protease inhibitor 450 and ombitasvir, which is co-formulated, and dasabuvir, which is a non-nucleoside polymerase inhibitor, has to be given twice a day. So if you use that regimen, it'll be 3 pills. And then you've heard about cimeprevir and sofosbuvir, decladisvir and sofosbuvir, and you have in phase 3 studies right now, esinaprevir, which is a protease inhibitor, along with decladisvir, and a non-nuc polymerase inhibitor. And so you'll have a 2-pill regimen there. So it could be 1, 2, or 3. And if you were to require ribavirin, you'll have to add anywhere from, actually should be 5 to 6 pills a day. So I will go over various treatment combinations just to give you all a little bit of flavor as to how well these work. Sofosbuvir and cimeprevir data I'll show, decladisvir, sofosbuvir, and then sofosbuvir, lenipasvir data, and then the 3-DA regimen, and then the 5172-8742 preliminary data, and some decladisvir, cimeprevir data. So you heard a lot about this study, the Cosmos study. It's really unbelievable that a study that has just over 160 patients has achieved such prominence. In fact, in our center, we have more patients on this combination than actually what they had in the study. So there were 2 cohorts of patients who were treated for either 12 or 24 weeks with sofosbuvir and cimeprevir with or without ribavirin. The first cohort was of patients who had mild fibrosis or no fibrosis, and these patients were, and they're null responders. And then you had second cohort of more advanced fibrosis, naives, and null responders. And you can see here pretty high response rates. What I want to point out here is that if you were to look at a couple of arms, there were 14 patients here, and 13 or 14 responded. If you had 1 patient not respond, this number would drop from 93% to around 87%. So if you have 2 additional patients not respond, it would drop even further. So everyone is excited because it's above 90%, but the confidence intervals in this study are going to be extraordinarily wide. That's why we don't even show you the confidence intervals here. Yet over 90% response rate is being seen. So we know that cimeprevir has a bit of a challenge against baseline QADK polymorphism in that based on the Quest-1 study, particularly those who had baseline QADK polymorphism did not do as well. And it is recommended that you test for QADK if you're going to treat treat the 1A patients. 1B, it's not an issue. And here in this study, based on QADK polymorphism in genotype 1A patients, you really didn't see any poorer response. And this may be because, 1, the number of patients treated in the study was relatively small. 2, sofosbuvir may overcome the weakness of cimeprevir to 180 QADK polymorphism. So, of course, this is an all-oral regimen, and you can see that it was well tolerated. It had some fatigue, headache, nausea. But if you were to look at rash, it was more in the ribavirin arm, about 15% and 13% here. Pruritus as well, particularly with longer duration of therapy in the ribavirin arm. Now, the next study that has achieved quite a bit of prominence as well is the decladisvir, NS5A, and sofosbuvir study with or without ribavirin. And you can see here, this was done in stages, and cohorts were added as they got data, and we got a little more confident. And we participated in this study as well. And initially, they had a lead-in arm as well of sofosbuvir before decladisvir was added. And then it was initially 12 weeks, and then 24 weeks, and then 12 weeks. Then it was extended to genotypes 2 and 3 patients, and then eventually 24 weeks of treatment for those who had failed telaprevir and boceprevir. And if you were to look at the response rates, extraordinarily high in these patients, as high as 100%. But then if you were particularly were to look at protease inhibitor failures, a relatively small number of patients in the 20s, those who failed telaprevir and boceprevir had a high response rate. So this is an option that would certainly be very viable for those of your patients who failed telaprevir and boceprevir. Genotype 3 patients had about an upwards of 80% response rate, but this is an overall response rate for genotypes 2 and 3. So well performed very well. So then with those small studies, let's talk about the larger phase 3 pivotal registration studies. So I'll present a couple of programs. One is of ledipasvir, an NS5A inhibitor that has activity against 1A and 1B, and sofosbuvir, which is pan-genotypic, the NS5B polymerase inhibitor. So there are 3 studies called the ION studies that have been done, and I will present each of these to some extent. The first study was ION-1, which had 12 and 24 weeks of treatment with ledipasvir and sofosbuvir, with or without ribavirin. And as you can see here, the response rates were extraordinarily high, 12%, 12 weeks and 24 weeks at upwards of 95% sustained virologic response. And ribavirin did not make any difference. So one could conclude from this that this regimen does well with just 12 weeks of treatment, and ribavirin is not necessary. Now, if you were to look at relapse rates, extraordinarily low, and keep this in mind because when you look at a shorter duration therapy, this becomes important. If you lost to follow-up, there was one who had breakthrough, and it was concluded by the investigators and the sponsor that this was due to non-compliance. If you were to look at adverse events, 12 versus 24 weeks, relatively few serious adverse events. And then if you were to look at treatment discontinuation due to AEs, relatively low. What is important to recognize is that hemoglobin drops are in the ribavirin-containing arm, less than 10 grams, 20 patients on 9% had in the 12-week triple therapy regimen, and it's here 17 patients had it with extended duration of therapy to 24 weeks. Now, ION-3 is another registration trial in treatment-naive patients but without cirrhosis. And here they tested the 8-week regimen of sofosbuvir and ledipasvir with or without ribavirin, and then they had a 12-week arm of sofosbuvir and ledipasvir but without ribavirin. Keep in mind, this is entirely a non-cirrhotic population. And again, you saw very high response rates of upwards of 90%. So if you were to look at these response rates, you'd say 8 weeks is as good as 12 weeks, and that ribavirin is not needed even with the 8-week regimen, and people are expecting that this regimen would be approved as an 8-week regimen without ribavirin. But this is where I have a bit of a challenge. If you were to look at relapse rates in the 8-week arm, there are 20 relapsers. So if people fail, they want to know why they failed. Nowadays, since response rates are so high, about 90%, people are getting greedy and want to know, hey, exactly why is it that few patients fail? So there are 20 relapsers in the 8-week arms, as opposed to 3 relapsers in the 12-week arm. There's actually a 5-fold increase in relapse rate with the 8-week regimen. And if I had a patient in front of me, I'm not sure what I'm going to say, whether they should go with the 8-week arm or the 12-week arm, unless we come up with a definite rescue schedule for these patients who failed the 8-week arm. I'd find it a little bit of a challenge to readily agree to that, but this can be discussed. There is actually a rescue protocol for these few patients who relapsed. Now, this is ION-2, which is an important study, and this is in treatment-experienced patients, 12 and 24 weeks treatment with or without ribavirin. And this allowed for telaprevir and boceprevir non-responders. Actually, there were a significant number of patients who had failed telaprevir and boceprevir, and in these patients, there were a significant number who had baseline resistance. And here you can see here, if you were to look at prior protease inhibitor failures, as high as 60% and as low as 46% of the population in the various arms were prior protease inhibitor failures. And you had about a 20% cirrhosis rate in this population. And again, you saw very high response rates, 12 and 24 weeks of treatment. But then when we're broken down by cirrhosis, presence or absence, the 12-week group did not do as well as the 24-week group, in that there was a low response rate of 82 and 86% with or without ribavirin. So one could use the strategy in a prior treatment experienced patient, but in a cirrhotic up to 24 weeks to optimize outcomes. Now again, here you can see the relapse rate was higher in the 12-week group as opposed to the 24-week group, and 7 out of these 11 patients were cirrhotic patients. So again, clearly, cirrhotics tend to relapse with a 12-week regimen in the background of prior treatment experience, and they should be treated for 24 weeks if you had this regimen available. I will now go on to the next treatment program, which is the 3 directly acting antiviral drugs, the ritonavir-boosted protease inhibitor, ABT-450, and then you have umbitasvir, which is an NS5A inhibitor. Those are co-formulated, and then you have dasabivir, a non-nucleoside NS5B polymerase inhibitor that has to be given twice daily. Now, there are several studies that have been done, and it's actually a very robust, systematic program that this combination has gone through. I will not belabor the point that has already been made that, sustained response rates, what happened here, have been extraordinarily high, upwards of 90% in any of these programs, and the patient populations varied. Genotype 1B treatment experience were looked at, 1B treatment naive, 1A treatment naive, 1 treatment naive with compensated cirrhosis, etc. Incidentally, these are all genotype 1 patients. So I want to just present this study, which is important because patients ask me, what kind of side effects can I expect from these drugs? Because people have been so afraid of interferon and ribavirin, they're expecting the same kind of side effects. Believe me, even if you have these medications, it's not easy to convince all patients to take these treatments because they've heard about the disastrous side effects of interferon and ribavirin. Other day, I had a patient, actually a physician, with genotype 1B, and I said, you got the new jackpot of hepatitis C, you can get rid of it easily. He said, well, you only treated 300-odd patients in the clinical trial, or 600-odd patients in cardiology. She's a cardiologist. They treat a few thousand patients before they can assess the efficacy and safety. So I want to wait a few years before I take this treatment. So it's not easy to convince everyone to take this treatment. So for that matter, this is an impressive study in that they had a placebo arm for safety and adverse event comparisons. And it was heavily weighted towards active drug, 473 patients who got active drugs as opposed to 158 who were on placebo. And This is basically the usual kind of breakdown that you see. And the sustained response rates were extraordinarily high in both 1A and 1B patients, and far superior to the superiority threshold that was established based on historical studies done with protease inhibitors. Now, if we were to look at a breakthrough, there's just 1 of 473 patients had breakthrough. There were 7 of 463 patients had breakthrough, relapse rather, and this was more in genotype 1A patients, which I'll not show. But this is what I want to point out. If you were to look at fatigue and headache, there was not statistically significant. Both the placebo group and the active treatment group had fatigue and headache. So when someone says, when you're taking the medication, I have fatigue and headache, you don't know if it's due to hepatitis C itself or due to the drugs. So hepatitis C itself can cause some symptoms. So and then this study has quite nicely demonstrated that not all adverse events or symptoms are related to active drug treatment. Of course, nausea, pruritus, insomnia, and diarrhea were more common in the actively treated group. Now, I want to then present this cirrhotic study, and again, my compliments to the investigators for doing this exclusive cirrhotic study, which needed to be done. If you look at the ION program, it had threshold of 20% cirrhotics in a couple of studies, ion 1, ion 2, but not ion 3. But here, this was a large study done in exclusive cirrhotics, over 380 patients. And if you were to look at response rates with 12 weeks or 24 weeks, you had comparably similar sustained virologic response rates over and 90% either with 12 weeks or 24 weeks. Now, this also, by the way, this study also had treatment experienced patients, and if you were to look at prior non-response and now response to 3-DA regimen in genotype 1A patients, the prior null responders did less well compared to with 12 weeks of treatment compared to a higher response rate with 24 weeks of treatment. So again, in a cirrhotic who has a prior null response to interferon and ribavirin, 24 weeks of treatment with triple DA might be the way to go. Now, if you were to look at 1B, that kind of difference is eliminated. You could see a high response rate. The prior partial responders had a low response, but again, look at the number of patients, 7 patients. So I'm not sure how I would interpret this data for just based on 7 patients. So 1B, as we all recognize, is less challenging to treat than 1A. I want to present a bit of data from daclatasvir and esinaprevir. It's a complex treatment strategy. You can see here there is daclatasvir and esinaprevir for 24 weeks. They're actually in treatment-naive patients. There's also a shorter duration 12-week treatment arm. Non-responders were treated with daclatasvir and esinaprevir for 24 weeks, and interferon ineligible intolerant were treated for 24 weeks with an NS5A inhibitor and a protease inhibitor. The response rates, before I go into response rates, you can look at the rate of cirrhosis in the non-responder and interferon ineligible intolerant. They ranged from 31 to 47%. As you can imagine, prior non-responders are likely to be non-CC. Most of them were non-CC patients. The response rate is here. In treatment-naive patients, is 90%. Non-responders are 80%. And this is where the challenge is going to be. Remember I said the treatment duration was 24 weeks. While the response rates are higher, it's going to be hard to kind of sell a 24-week regimen when 12-week regimens have done much better than these response rates. Yet, this is still an option for our patients. Then I'll show some preliminary data with the protease inhibitor 5172 and the NS5A inhibitor 8742. They're 12 and 18 weeks treatment regimens. Initial cohort at treatment-naive cirrhotics, and then the cohort had PEG ribavirin null responders and cirrhotics or without cirrhosis. And you can see here, this is the data in evolution. It was presented recently. You can see response rates that are quite high, follow-up though only for 4 to 8 weeks. And you can see here, their patients more often had relapse, the ones who failed. There was one breakthrough, but overall very good response rates with this combination treatment. And this is now in phase 3, it's due to start phase 3 pretty soon. Now, I want to present an interesting study that serves as a proof of concept that perhaps we can shorten the duration of therapy with the addition of more DAAs. This is the so-called Synergy trial done at the NIH, where they had 3 treatment arms. One was a sofosbuvir and lenacapavir for 12 weeks. Then they had a sofosbuvir, lenacapavir, and they added another DAA, which was a non-nucleoside NS5B inhibitor. And then one arm received additionally a protease inhibitor of the sponsor, GS9451. And they treated treatment-naive patients, all stages of fibrosis, one arm, 20 patients with sofosbuvir and lenacapavir. They treated treatment-naive patients with excluded cirrhosis, but it was just 6 weeks of sofosbuvir, lenacapavir, and GS9669, 20 patients, and then treatment-naive patients, cirrhosis excluded, with another DA along with sofosbuvir and ribavirin. And this was done in an inner city area where there were a good number of black patients, non-CC patients, and they had sustained response rates of 95 to 100%. The only one patient failed in the sofosbuvir, lenacapavir, GS9669 arm. So this is, again, impressive enough for us to consider such treatment as we move along. Briefly, I want to touch upon genotypes 2 and 3, and most of it is going to be with sofosbuvir and ribavirin. The first proof of concept came from the electron study, where you saw patients with sofosbuvir and ribavirin have 100% SVR. What is interesting is there was an arm where they just used sofosbuvir, and there was a 60% SVR in this cohort. And of interest is that all those who are genotype 3 actually responded to sofosbuvir monotherapy. But again, this is a very small study. Now, the approval of sofosbuvir and ribavirin for genotypes 2 and 3 is based on these studies, the fission study, fusion study, and the positron study. Well, for treatment-naive, treatment-experienced, and intolerant patients, 12 weeks of sofosbuvir and ribavirin in the fission study compared to 24 weeks of standard of care of interferon and ribavirin. And then you had 12 and 16 weeks treatment for the treatment-experienced, and then just a single-arm study for the interferon intolerant. And I presented this yesterday. Generally, genotype 2 patients have done very well. The challenges with genotype 3, as you can see here, genotype 3 patients in the treatment-naive did better with PEG, interferon, and ribavirin for 24 weeks. That's a numerical difference, not a statistical difference. But in the treatment-experienced patients, the 16-week arm did better than the 12-week arm, and this was statistically significant. And in the interferon-intolerant patients, sofosbuvir and ribavirin for 12 weeks had a 60% SVR. So genotype 3, there are low response rates, and this was overcome by extending treatment duration to 24 weeks in the Valence study, although the cirrhotics who had genotype 3 and were treatment experienced did not do as well with 60% SVR. So what about future prospective regimens for genotype 3? One is of decladisvir and sofosbuvir. In treatment-naive non-cirrhotics, 24 weeks of treatment had an 89% SVR, but you have to be careful with the number of patients, only 18 patients in this cohort. Or you could look for the next pan-genotypic NS5A inhibitor, GS5816. This is entering phase 3 right now, but this is the phase 2 data where they looked at 25 milligrams and 100 milligrams of GS5816 along with sofosbuvir in genotype 1, 3, 2s, and 6s. And you can see here extraordinarily high response rates in these patients with 100 milligrams. In fact, in the phase 3 study, it is going to be 100 milligrams of GS5816 co-formulated with sofosbuvir, and it will be a pan-genotypic treatment regimen. So hopefully and potentially, if this works, you can eliminate the need for genotype testing. You could just give 12 weeks of treatment for any genotype and achieve high response rates. So just to give you a little bit of sense about the EC50 of the various NS5A inhibitors, you'll see that GS5816 has a very good EC50. Ledipasvir is a challenge. You can see here the EC50 is not good. Decladisvir is quite good. H742 is quite good as well. And you have other drugs as well. So ledipasvir is not to be used for genotype 3 patients. Decladisvir certainly is an option. So as we move forward, this is how I look at it when it comes to treatment options in the future for hepatitis C. There's got to be a drug that has to be a platform. And you'll have various arguments about this in terms of what should be a platform. One could argue that it should be an NS5B nucleotide polymerase inhibitor, and then you can partner it with any of the other drugs, an NS5 inhibitor or a pan-genotypic protease inhibitor. Some might argue that it's an NS5A that is the platform for combination treatments. And I can assure you, those who are working with the ritonavir-boosted protease inhibitor will argue that for genotype 1, a protease inhibitor should be the platform. So you could use a variety of combinations. And then the treatment duration, is it going to be 6 weeks, 8 weeks, 12 weeks? Is it going to be different for 1A and 1B? Some regimens are going to be different because the 3DA regimen without ribavirin performed extraordinarily well in 1B, but 1A, there were few more relapses. So 1A, you might need a ribavirin. Cirrhotics, we recognize that we need longer duration of therapy, particularly the treatment-experienced patients. So race and ethnicity, I think, has been overcome. I don't see a need for IL-28B status as we move forward with these drugs. Viral resistance might be issues when you're trying to expose them to repeated treatments, and then we've heard extensively the challenges with regard to the cost. So at least in late 2014 and beyond, I see this. For genotype 1, there'll be 2 approved regimens, sofosbuvir, ledipasvir, and the 3DA regimen. Of course, if you have decladisvir, you could use that along with sofosbuvir, but these would be the approved regimens. Now, in Japan, decladisvir and acitonopravir is approved for genotype 1B. For 2s, I don't think you're going to do very much better than sofosbuvir and ribavirin, at least for the immediate future. And genotype 3, you have sofosbuvir and ribavirin, but a preferred option or for some might be pegylated interferon, ribavirin, and sofosbuvir. So at this stage, I'll end, and I thank you very much for your attention.